Immunity is the processes that occur to defend the body against foreign organisms or molecules. 2. Immunity includes A. Inflammation, B. Complement activation, C. Phagocytosis, D. Antibody synthesis, E. Effector T lymphocytes. Types of immunity. 1. Innate, nonspecific or natural. A. Born with it, do not need prior exposure. The effectiveness of the immune response varies with age. C. First line of defense, designed to keep microorganisms out. 1. Physical barriers such as epithelial cells, intact skin, trapping of bacteria in mucus, etc. 2. Chemicals secreted by cells and tissues such as acidic pH of skin surface, complement, interferons, lysozymes, etc. D. Second line of defense. 1. Phagocytosis, the process of a white blood cell WBC engulfing bacteria. 2. Inflammation, nonspecific response to tissue damage that includes A. Chemical release, B. Cellular movement, C. Elimination of foreign material, D. Tissue repair. 3. Complement system, enhances phagocytosis, stimulates inflammatory response and lyses foreign cells. 2. Adaptive, specific or acquired, A. Acquired only after a specific challenge is encountered and responds specifically to the challenge. B. 2 responses. 1. Humoral-mediated immunity, HMI, A. More important in protection against extracellular pathogens, B. Antibody production by plasma cells. 2. Cell-mediated immunity, CMI, A. More important in protection against intracellular pathogens, B. Natural killer NK cells, some activity against tumor cells, C. T helper cells, D. Cytotoxic T lymphocytes, CTLs, E. Cytotoxins. Active immunity. 1. Natural, the host is exposed to foreign immunogen as a result of infection, and the host's immune cells manufacture specific products to eliminate foreign immunogen. 2. Artificial, vaccination. Immune system responds to an altered, non-infectious organism. 3. Active immunity generally endures for life. D. Passive immunity. 1. Natural, maternal antibody crosses placenta to protect infant. 2. Artificial, immune products from another animal injected into the host, e.g. pooled gamma globulin. 3. Passive immunity, short-term, no memory cells produced, E. Antigens and immunogens. 1. Immunogen, a substance capable of inducing an immune response. 2. Antigen, a substance that specifically interacts with cells or substances of the immune system. Immunogens are also antigens, but not all antigens produce an immune response. 3. Epitope, the portion of a molecule, i.e. antigen, that binds to an antibody or T cell receptor. 4. Thymic-dependent immunogens, molecules that require T helper cells to stimulate antibody formation. 5. Thymic-independent immunogens, molecules that initiate antibody production without stimulating T helper cells. 6. Immunogenicity characteristics. A. Foreignness, must be recognized by the body as non-self. B. Size, greater than 10 kilodaltons. C. Chemical composition. Proteins and carbohydrates are the most immunogenic, whereas lipids and nucleic acids are weakly immunogenic. D. Complexity. The more complex a molecule, the more immunogenic it becomes. E. Route of entry into the host also determines immunogenicity. F. Dose of immunogen affects immunogenicity. G. Degradability. The immunogen needs to be degraded and presented to cells of the immune system. 7. A hapten is a low molecular weight molecule that alone is too small to stimulate an immune response but can combine with another molecule to induce a response. 8. Adjuvant is a compound that enhances an immune response. It is not immunogenic and cannot induce an antibody response alone. F. Antibody, immunoglobulin, Ig or gamma globulin, is a protein that binds to antigens. There are 5 classes, IgG, IgMe, IgA, and IgD. Antibodies primarily migrate in the beta and gamma regions during protein electrophoresis. 1. Antibodies are composed of 2 heavy polypeptide chains and 2 light polypeptide chains. A. Light chains I2 types, kappa and lambda. 2. Antibodies have only one type of light chain, kappa or lambda. B. Heavy chains. Immunoglobulin classes are defined by a unique heavy chain, IgM mu, IgG gamma, YGA alpha A, IgD delta, IgE epsilon, E. 2. Every heavy chain and light chain consists of one variable domain and one or more constant domain. 3. The variable domain defines the specificity of an antibody. This portion of the molecule is referred to as the fragment of antigen binding, Fab. 4. The crystalline fragment, Fc, of the antibody is located at the carboxy terminus. It is responsible for the biological activity of the molecule, including activating complement. 5. Antibody heterogeneity. A. Isotypes, variations between light and heavy chains. E. Defined by constant regions of all antibodies and kappa and lambda light chains. B. Allotypes. I. Species-specific variations in the constant domains of heavy or light chains. E. different alleles of heavy and light chains. C. Idiotypes. I. Variation in the variable region. E. A single clone of cells produces a single idiotype. 6. J. Joining chain. Multiple monomers of IgM and IgA are linked by a J chain. One J chain is needed for each IgM or IgA molecule that is linked together.